Hello everyone, my name is Alexandre Antonelli and I'm the Director of Science at the Royal Botanic Gardens Kew. And it's my great pleasure and honor to welcome you all to the 2023 State of the World's Plants and Fungi Symposium. And I would like to start by thanking the Sfumato Foundation for generously funding the State of the World's Plants and Fungi project, and the exhibitors and advisors for the support, the New Phytology Foundation, Plants People Planet, Anazal Botany, and Botany One. I'm pleased to say that we are about 250 in-person participants here in the beautiful Kew Gardens in West London in UK, and about 1,500 online participants joining us, as well as people joining online on our free YouTube channel. So this is a truly international symposium, and you're going to see that in the next 6 thematic sessions and the 4 workshops over the next 3 days. But before I talk more about the specific contents and the purpose of the symposium and the report, I would like to invite the Right Honourable Lord Benyon, who's Minister of State at the Department of Environment, Food and Rural Affairs, and the Minister responsible for Kew.
Though I can't join you today, anyone who knows me will tell you that whether I'm speaking in Parliament, whether I'm at a global summit or one of our diplomatic missions around the world, I will always take the chance to wax lyrical about Kew for as long as I've got. It's hard to exaggerate the importance of Kew's work. Kew reports have shown that 2 of every 5 plants are threatened with extinction. It's such a horrific statistic that I'm going to say it again. 2 of every 5 plants are threatened with extinction. And this week we've learned that as many as 3 in 4 of the planet's undescribed plant species are in the same boat. But thanks to the dedication of over 200 scientists in more than 100 institutions across 30 countries, this latest series of publications unveils many more worrying revelations about the patterns of loss for plants and fungi. And these publications show encouraging insights into the progress achieved despite the challenges of the pandemic, and also new advances, including the latest in DNA sequencing, with the potential to help us turn the tide. And what comes across most clearly is that time is not on our side, and that Kew's work and that of others around the world is invaluable in helping us understand and communicate the need for urgent action to protect and conserve diverse populations of species of plants and fungi on which we all depend. The good news is that at the UN Nature Summit, CBD COP15 in Montreal at the end of last year, 196 countries came together to agree a shared global mission to halt and reverse the loss of biodiversity, with targets to protect at least 30% of the world's land and ocean by 2030. Behind the scenes, Kew did so much to highlight the role of plants and fungi. We were helped immensely by the way we worked together to put nature at the heart of our response to climate change at COP26 in Glasgow, supported by unprecedented public, private sector, and philanthropic investment and communities. And I should just say that it was so superb to see Kew front and centre of COP26, and now hardwired into future fora of that kind. And your level of expertise and commitment is recognised globally, as I say. So here at home, there is a vast amount of work underway to meet our commitments, including new environmental targets that we've now put into law. The UK is championing global efforts to get the market moving in the right direction and to meet our shared global commitment to get $30 billion a year for nature flowing from all sources into developing countries by 2030. I'm proud that the UK has made one of the first financial pledges to the newly created Nature Fund, with an initial £10 million to help it open as soon as possible. But this latest report reminds us how many astonishing secrets we are yet to discover. So I'll finish by wishing you the best for the rest of your symposium, and to urge you all to keep at it, to protect the web of life on our planet, and to make sure people everywhere have the food, the medicines, the materials they need to thrive for generations to come. And in all our endeavours, in all that is talked about in and around government and in public life and everything else that you hear in the froth of media attention, what you are doing here is much more important. It's absolutely fundamental to our future on this planet. I can't thank you enough, and I wish you well in the discussions you have over the next day or two. Thank you.
Thank you to Lord Benyon for this speech and support. So for those of you who may not be familiar with the State of the World's Plants and Fungi project, so the report and symposium, this is a symposium now that is taking place today and over the next couple of days, following the release on Tuesday of the report, the State of the World's 2023 for Plants and Fungi. And it's the fifth in a series of Q's State of the World's reports, which provides a digest, a regular digest of our knowledge about plants and fungi and also the threats they face. So the report is based entirely on scientific research. So there is a co-release of a special collection in the New Phytologist and the journal Plants, People, Planet for all the plant-based research underlying the report. And then there's also a global review of fungal diversity in the Annual Review of Environment and Resources. So it's about 25 publications, which then have been summarized in a very accessible way to anyone who may not be an expert in the topic, but hopefully will be able to really understand and engage with the topic of plant and fungal diversity and threats. And this year's report is focused on what we call in the nature emergency. And that's the language that governments and societies are using now, because it is an emergency. We need to face it. And how do we tackle that? We are trying to provide the best available evidence, but also highlighting the gaps and the priorities moving ahead in terms of understanding of plant and fungal life on this planet. And even though, of course, we want to focus on what we know, we also need to acknowledge and understand what we don't know, but we should know. And that's where the prioritization and moving ahead comes in, and I'll come back to that in a second. So the report itself is, of course, freely available for download anywhere. There'll be a number of copies here for attendees who are interested in having a physical copy. But we do encourage people to download and use the digital copy, of course, for environmental reasons, and also to spread in your networks. So it is available from that website, and the collection is available from the New Phytology and Plants, People, Planet special collection, available on the website you see here on the screen. And also for the review paper on fungal diversity and threats at the last link here you see on this page. And as Lord Benyon said, it is not a single person's effort. It is a truly collaborative international effort, which took about 2 years in the making. So about 200 scientists in over 100 institutions and 30 different countries have come together to generously offer their knowledge, expertise to produce the work that you'll be reading here in this report. So I'd just like to walk you through some of the highlights and the most exciting news and findings that the report conveys. So one of them is the fantastic celebration of the work of Raphael Kovacs, who may be here, sitting over there. And Raphael had a dream as a young person, and he really wanted to have a very simple list of all plant species on the planet and where they are. And so did Darwin, and Darwin himself, before he died, he really dreamed of a list of all plants on this planet and where they occur. And it took Raphael and his team about 35 years in the making of really dedicated work to come up with a list which we can now celebrate and use and that's a list that currently has about 350,000 species of vascular plants. Those are all the plants people are familiar with, except for the mosses in this case and algae. And we are now able to use this list for many different things, and that's what we're going to see as well as outcome in the report. Another really fantastic finding is the realization that we are discovering new plants and new fungi every single day. So almost every day, about 20 new plants and fungi are described as new to science. So since the last report came out in 2020, 18,800 species of plants and fungi have been described by the scientific community around the world. And there's some incredible species, not only those tiny things that perhaps no one has seen or will ever see. It's also one of the largest plants we have. That's the giant water lily you see in front of you from Bolivia. And it has the largest of all giant water lily leaves. And it's been undiscovered under our nose for about 200 years until horticulturists, scientists, a botanical illustrator and Bolivian partners came together to really describe this as new to science only last year. So that's just one of the many examples, and some of them which are absolutely incredible, and you'll read about them in the report. We are now also using some really advanced methods to better understand the patterns of diversity we see. So the report is very heavily based on the list, so that's what we use. We asked all the authors of the scientific papers to use the list to explore many different aspects of plant diversity and distribution, the evolutionary history, but also the conservation aspects and so forth. But we're also now using new technologies to help us integrate across different data sets and different techniques. So you'll see here, for instance, a snapshot of work that highlighted that about 80% of all biomass in the Northern Hemisphere in Europe is only accumulated by 3% of all the diversity. And how does that compare, and what's the implications for conservation and sustainable use of natural resources? Some of the works in the report and the papers are quite focused on different branches of the tree of life, so different clades such as orchids, one of the most species-rich of all flowering plants. And one of the findings there is that orchids, they lived together with dinosaurs about 83 million years ago. There were orchids on this planet. And the origin of orchids, although some previous research had suggested it may come from Australia, is here now estimated to be from the Northern Hemisphere. So those are just two of the findings that I'd like to highlight, but there are many in the scientific papers. And I really encourage people to, of course, read the report, but do go to the original sources as well of the paper so you can learn more about the different studies and different groups of plants and fungi as well. Another thing I'd like to highlight here is that it's not only about the numbers of species that matters for conservation, but also the fact that there are particular regions in the world where we have a very high concentration of undiscovered species. And those are what we're calling the dark spots. It's a bit sci fiction here, but the dark spots are really the places where we believe there will be the highest number of new species to science. In this case, plants, because that's where we have the best information so far. But basically, the new species and also the new locations, so the new geography. So species which was known from elsewhere, but is found in those places. And that's a really important aspect, because as I'll come to in a moment, we need to understand where to prioritize our research and conservation efforts, and also our collecting and training. And that's what the workshop is going to deal with in a moment. And there is very much still to do, and it's a very exciting time to be a biologist. Hopefully many of you will have students or will be able to communicate to scientists or colleagues about all the amazing things you can do as a biologist, not least in taxonomy, which is the science that documents and understands the distribution of plants and fungi. I also want to highlight a study here where the national species, so the fact that many species are only found in one particular region, one country or botanical country, is quite striking and something that people should be really proud about. You know, you come from a particular region and those species are not found anywhere else, and how we can champion that and really hopefully convince our leaders to invest the appropriate resources for protecting that diversity. So you see here that about 221,000 species are only found in one of those botanical countries as we define in the work checklist of vascular plants. But as Lord Benyon said, it's not only about good news and the fantastic discoveries we're making, but it's also about the really serious situation that we're facing today. And this report provides a new estimate that almost half of all plants, all flowering plants, are very likely to be threatened. So 45% is, I believe, in my personal opinion, absolutely a disaster. And it's something we have to take extremely seriously for the coming generations and for our ability to preserve life as we know it and our own future existence on the planet. So conservation is something that we all benefit from. It's not something that only matters to biologists. And I'm very pleased here that there are so many representatives, not only from scientists and scientific organizations, but also NGOs from different companies as well, and governments, because we all need to work together in terms of understanding and protect biodiversity for the future. I've talked a lot about plants, and plants are absolutely wonderful. They give us food, they give us fiber, building material, but there's another dimension of life on this planet which is absolutely outstanding, and it's fungi. And fungi are really the next frontier of biodiversity research. And I'm very pleased that you will have a tour in the lunch in our fantastic fungarium, so that's our collection of fungi. And we here provide a new estimate of how many fungal species there are on this planet. And this number is quite unbelievable. It's about 2.5 million species, which is the best estimate. It's between 2 and 3, considered all the information we have on molecular, so DNA-based studies, as well as classical taxonomic work based on specimens. But the issue is that we've only discovered less than 10% of those. So more than 90% of all fungal species, despite all the amazing things they can give us in terms of, again, food and medicine, and so many other really interesting traits, some fungi that eat plastic and break it down, and others that accumulate heavy metals and can help us clean our environments. Most of those species remain absolutely unknown to science. And on top of that, we have a very few species which have been formally assessed for their conservation or extinction threat. So that number, 0.4% of all species that have had their assessment in the IUCN, so International Union on Conservation of Nature, the red list, which is absolutely fundamental to putting the needed resources for conservation. So unless you have that categorization, that formal classification that you're threatened or not, it's really hard to get the necessary resources for conservation. So there are many different opportunities, I think, and when it comes to biodiversity and conservation, we do believe and we know that there is still chance to turn things around. And I think all the available evidence, Lord Benyon mentioned the Conference of the Parties, the COP15 meeting that the United Nations organized in Montreal last year. I was very fortunate to be part of those discussions and to see that despite all the differences and inequalities that exist on this planet, we are still very confident that if we put in the right resources and the right measures, we can still bend the curve on biodiversity loss. And this report is going to be fundamental to that, because we know that we cannot do conservation, we cannot use nature sustainably unless we get a better understanding of global and fungal diversity and the threats that it faces. So I'm really looking forward to lots of thought-provoking discussions here. There's a huge amount of expertise in this room today, so please do take the opportunity to have those discussions and interact with your colleagues. I also want to clarify that this symposium here is very much about discussing the themes at a broad level presented in the report. So please, again, do have a look at the report so you can familiarize yourself. But of course, speakers have been invited to contribute into putting the findings into a broader context of their own research and other initiatives taking place around the world. So it's a very inclusive, hopefully, way of discussing this topic at the different and far-reaching implications of conservation. And how do we fill the knowledge gaps ahead of us? Because it's, on one hand, fantastic to be able to celebrate the release that Raphael and his colleagues, together with many different scientists around the world, the list and accomplishments that we have made so far, but we also know that there's much we still need to know to really be able to protect all life on this planet. And in terms of outputs, I hope that this will hopefully give a lot of new information and inspiration to all of you participating here online at Kew Gardens, but also everyone who's online and is able to join us and listen to the discussions. We also, of course, want to raise awareness for plants and fungi and celebrate them because they're so, so important. And please, many of you are here perhaps officially on behalf of your organizations. Do take this opportunity to bring in different perspectives. I was talking just before coming in here with people from industry, for instance, and really interested in the possibilities of using the traits that species provide us, and how do we do that complying to the international regulations? How do we do that in a really inclusive way that benefits the different parts of this world, Global South and Global North? Another thing we want to do is to start now an agreement for international cooperation to fill the critical knowledge gaps. And we have 4 workshops, and the idea is to draft a declaration which will then basically gather the commitments from organizations in terms of where and what to study in the coming years so that we can help delivery of the Global Biodiversity Framework. So the agreement that was signed by almost 200 countries, as what Benyon said. And it's really important that we don't spread our resources too thinly, that we are very efficient and ambitious, but also that we really join forces and create more synergy than the sum of the different parts. And I think that there's some really good opportunities for organizations to come together and work with local, regional partners in order to really understand and classify and really map our plant and fungal diversity around the world. So hopefully many of you will be compelled to participate in those discussions in the declaration workshops. So in terms of setup for this symposium, I hope you had a chance to look at the website, what the plan is. But today, we're going to have the first session, which will focus on the diversity hotspots and critical knowledge gaps. And then I really hope everyone will have the chance to look at the posters. There's some fantastic contributions I've seen of students and professionals presenting some of their work in relation to the topics discussed in the symposium. So please do take the time to see those posters, but also interact with the students and scientists who have designed them. On day 2, which is tomorrow, we're going to continue talking about the diversity hotspots and critical knowledge gaps. We're covering both plants and fungi in those discussions. And we also focus, importantly, on what we know about extinction risk. And it's really important that we talk about extinction. On Friday, the 3rd day of the symposium, we'll have a session on how do we accelerate our understanding of plant and fungal diversity. What kind of methodological advancements, new techniques we can do to do that, because otherwise it will take centuries for us to document every single species of plants and fungi. So we need to understand how we can use a combination of expertise, collections, but perhaps also new advances in DNA technologies, artificial intelligence and so forth, to really help us to deliver that. And finally, a session on how do we achieve global and national targets for biodiversity protection and restoration based on the understanding we have of plant and fungal diversity. So we'll be summarizing the points of discussion across the different workshops. There'll be scribes who have very generously offered to take notes and get the main ideas so that we can use those for the declaration. And we also have a discussion, which is going to be very inspirational, I'm sure, about how to make biodiversity research count. So how do we win hearts and minds, and how do we drive change by a professional communicator? And also, we'll have a Posters Awards and closing remarks at the end. So many things happen over the next 3 days, which I hope everyone will be very, very excited to participate in. In terms of aspirations, I really hope that there'll be lots of different perspectives being brought together. So there's participation from many different countries worldwide. We are also keen to strengthen the links to policy and on-the-ground impact. So we're always having that in mind, that it's not only an exercise we're doing in an academic bubble, but it's actually a real world out there that we are trying to save and help protect. And again, it's very much in line with the global commitments that have already been made in particular the Kunming-Montreal Global Biodiversity Framework agreed last December. There are many people online, and I really encourage everyone to be participating actively. So I'd like to ask those of you online, looking at the cameras over there, to please use the Q&A function to ask any questions. You can also use the chat for more general discussions. And of course, please, again, engage thoughtfully so you contribute pertinent thoughts and ideas, but always be respectful of other participants at all times. And I should also say that we are very keen for this to be as inclusive and collaborative as possible, so we also encourage everyone to go online and follow us on X, previous Twitter, use the hashtag State of the Words Plants and Fungi, so the abbreviation there, SOTWPF, and @QScience. And in this way, we can also engage with even broader audiences, so please do become active if you can. I just want to finish here and say that this is going to be a really fantastic symposium. I'm delighted that so many of you could make it here to beautiful Kew Gardens today. I hope you'll have a chance to really get to know many people who you haven't met before. So do spend time with those you don't recognize. And I think that there'll be some really good opportunities for collaboration, for interactions, which will go beyond the symposium as well into next years and so. And with that said, I'd like to now offer all of us to jump straight into the first session, which is going to be, where are the diversity hotspots and critical knowledge gaps? So without further ado, I'd like to invite Dr. Iain Darbyshire, who's going to chair the first session of this afternoon. Thank you so much, everyone. Thank you.
Hello, everyone. Welcome to the first session of talks. And as Alex said, we're focusing on the questions, where are the diversity hotspots and critical knowledge gaps? So these are central themes for this symposium. And this is going to be spread over 2 sessions, the second one being tomorrow. So we're going to have a range of talks over those sessions, and they're going to span from global or regional perspectives and syntheses of our knowledge of plant and fungal diversity and knowledge gaps through to country-specific case studies. Just to reiterate the announcement that delegates joining online can ask questions to the speakers using the Q&A function on the symposium platform, but those joining on YouTube or on Zoom won't have access to that Q&A function. So each speaker will have a short period of questions time at the end of their talks, and then there will also be a panel discussion in tomorrow's session 2. Okay, without further ado, our first speaker is Dr. Tim Utteridge, who's been leading research botanist at Kew since 1999, specialising in the flora of Southeast Asia. Tim is about to take up a position as Director of Botanical Research at Singapore Botanic Gardens in the very near future, and that's Kew's loss and Singapore's gain for sure. He's speaking today on plant diversity of Indo-Australia, understanding knowledge gaps from Thailand to New Guinea. Thanks, Tim.
Thanks, Iain. Hi, everyone. My name's Tim Utteridge. I'm a botanist in the Asia team here at Kew, and I'm going to be talking about plant diversity of Indo-Australia. So many of you might know that as Southeast Asia or the Malay Archipelago or something like that. But there's a general view that if we use Indo-Australia, we can actually be more inclusive of a greater part of the region where the Malay peoples are really those from Peninsular Malaysia and parts of Borneo. So, for example, on Twitter, quite a few of Indonesian botanists saying, you know, let's have a broader term here, let's use Indo-Australia, and that includes a lot more communities from that part of the world. So where are we? So we're talking about the biogeographical region of Malaysia. That starts from the Peninsula Malaysia, goes all the way through to the edge of Papua New Guinea, encompasses several countries, the largest of which is Indonesia, stretching all the way from Aceh all the way to the border with Papua New Guinea. Now, this area really came to prominence with Wallace's book, Malay Archipelago, which was published in the late 1860s. And of course, Wallace wrote the letter to Darwin that spooked Darwin to publish Origin of Species a little bit earlier than he wanted to. Wallace's letter was read out with Darwin's sort of introduction at the Linnaean Society. And Wallace drew one of many lines on the region, and we have a tangle of lines through the region now, mainly which conform to different limits of animals. So things like elephants, tigers, rhinos, only found in the western part. The middle, very deep, geologically quite young. And then on the far eastern side, we have Australia connected to New Guinea. And during the Pleistocene, when the ice caps sucked all the water up and sea levels went down, we were able to walk from Asia to Borneo, and you could have walked from Australia to New Guinea. So let's have a quick look at these regions. Far west, we have Sundaland, the Sunda Shelf, dominated by the islands of Borneo, Java and Sumatra. We have things like woodpeckers, trogons, orangutans, elephants, rhinos. These don't go east. And plant-wise, this area is dominated by the Dipterocarpaceae, so a family well named because they have two-winged fruits, Dipterocarpaceae. These are victim of their own ecological success. They are massive trees. The tallest tropical tree is a shoria from northern Sabah, over 100 meters. They have straight boled to the canopy, and they have been logged and formed the basis for the timber industry in the early 20th century. And much of the lowland forest that would have been dominated by dipterocarps has now been converted. In the center, we have a region which is now called Wallacea, and here we have a mixture of organisms, so some unique animals like the babirusa. Halmahera, we have the first bird of paradise, so this is Wallace's standard wing in an endemic genus just found on the islands before we get to New Guinea. And I've shown a picture of a eucalyptus here, because in the southern part of this area, we have a lot of seasonal forest, and this is still poorly known. And when we get to the east, we have marsupials. And so we have tree kangaroos, wallabies, birds of paradise with about 45 species. And then the plants themselves, we have things like the clove families, Isidium. They've gone crazy. Bananas, center of diversity for wild bananas in New Guinea. And then in the bottom there, we have the epacrids, the southern heaths, members of the Ericaceae family, which is radiated, and the Ericaceae themselves have radiated in New Guinea. But if you're traveling through Southeast Asia, unfortunately, this is some of the habitat that you will drive through for many hours rather than driving through dipterocarp forest. This is palm oil. This is oil palm. This is a very productive oil tree. Within 4 years of planting it, you can get crops. Very efficient. If we want vegetable oil in the quantities that we do and we convert it to something else like coconut, we would need a much larger area. So the key is, yes, let's grow some oil palm, but let's be efficient how we manage it, and let's not waste what we've got. So if we look at the region, and this is WRI's intact forest landscapes, and these really show areas where there's minimal human impact on some of the forests. And many of these will match up to some of the dark spots that you'll see in the report, such as Borneo, New Guinea, Sumatra. And as a consequence of that, many of parts of Southeast Asia, or the Indo-Australia area, were identified as hotspots by the CI paper. Obviously, New Guinea wasn't, because it hasn't suffered 70% forest impact on its forest. So this was identified as one of the wilderness areas, which CI put forward as a joint conservation effort would really conserve large areas of the planet. So there was New Guinea, Miombo woodlands, Congo forest, Amazonia, and then just underneath southern US and Mexico. So in our part of the world, we're served by floras. Taxonomists write floras, they write descriptions, they form checklists, but we have no complete flora. Flora Milesiana there in the middle, that is been going since about the 60s, perhaps only 30% complete for a flora of about 45,000 species. Many of the countries and herbaria have started their own floras, and probably the only one that will be completed is the flora of Thailand, which is likely to be finished in the next 2 years. And that's been a massive effort by the Thais to really embed PhD students, herbaria, etc. since the 60s. And that's a flora of about 12,000 species. These are data from 2017, and obviously we could have searched, got the more up-to-date one, but this is from a paper where we looked at how the floras are getting on. And you can see that all of the regions, about 400 new species over a period of about 5 years. So still a lot of species being described. And recently we did a checklist, a taxonomic critically checklist of the island of New Guinea, and 99 authors, many of you will be in the room. But what I've highlighted there in green is we're talking about gaps in plant diversity knowledge. And what we're really looking at there is gaps in plant diversity capacity to do stuff. There are 5 New Guinea authors on that list out of 99. And that really sums up the state of herbaria, universities, and botanists on the island of New Guinea. So yes, we're talking about areas which need more exploration, but they need more capacity to do their work. So New Guinea came out as very rich. It's the second largest island on the planet, so over 13,000 species. New Guinea itself is very complex. Australia has smashed into it. It's risen up to nearly 5,000 meters. There's small glaciers on the top. We have large mangroves areas in the south, some of the largest mangrove trees in the world, rising up lowland forest, huge expanse of cloud montane forest going up to subalpine, rhododendrons, all the way up to forests at the top, which go into lichen and alpine scree. And you can see that new species are still being described. But if we look at the checklist that we did, we took all our to-do lists and our things that we didn't know, and we estimated about 4,000 species to describe. And if we think we're doing 400 every 5 years, this is another 50 years' work at least to finish off this inventory. And there's really there's many ways to discover a species. Obviously, you go collect, find something you've never seen before, such as this Pittosporum described from Angie Lakes, and this is going to be one of the first tropical important plant areas designated by Kew, over 60 endemic species from this part of New Guinea. So we go and we find new species that's never seen before. Yes, we can describe that as a new species. But there's also many hidden species in floras and revisions already done, and these are only revealed with more field work and more examination. So this is a small example, one of the most common trees in New Guinea plots, Medusanthera laxiflora, found throughout lowland New Guinea along the northern coast of New Guinea. And this was a very variable species with fruits that were dispersed by all sorts of different birds. And when we went and did some work in southern New Guinea, we saw that there's several populations with these large pink golf balls-like fruits that fall onto the ground. They don't remain on the tree. And once we looked into this and the variation and the population size, we're able to extract these species, and these are cassory dispersed species. So cassories are almost taking over a herbivory role for terrestrial animals in Australia and New Guinea. So from this field work and this ecological understanding, we've split that species up. And of course, the great frontier for herbaria is now next generation sequencing and being able to basically get DNA out of any specimen. So specimen level phylogenomic work is going to really increase our knowledge of variation, natural selection, evolution, and then understand the number of species there. So this is a very common species. This is Mesa haplobotris from Australia all the way through to New Guinea. We sampled the genus Mesa that I did my PhD on, but 20 years ago I really didn't do any NGS. And we found that there's at least 10 species coming out in different places in the tree, all called this name. And this is going to happen again and again and again in many groups, where species that have already been treated in floras and revisions are going to be split up into many different species. So where should we go? This is a map of the digitized specimens from New Guinea. So this is from like GBIF and other places. And these white areas, they have no digitized specimens. But I've been working on the island for many years, and there are no specimens there to digitize. So they haven't been collected. So some of these areas need to be collected very quickly. The red patch over here, this is next to the Lorentz World Heritage Site, the largest protected area in Southeast Asia, with practically 0 plant collections from that area. And immediately west of that, it's very flat. It's being converted to oil palm as we speak. There's the new Trans-Papuan Highway going through there. And we need to focus ourselves. This is our project in southern New Guinea. Botanists love going to see pretty plants, who love climbing mountains. And we went to the Alpine region and we kept going there. Look at the last 4 expeditions. Cost thousands of pounds. We added about 10, 15 species to the species accumulation curve. It's a bit rough, but you know, it does what it says on the tin. We need to go to places that are under-collected. And if you look at New Guinea as a whole, southern rainforest, 5.6 collections per 100 kilometers, and the other one, the freshwater swamp forests, I mean, difficult to go there. Again, about 6 collections per 100 square kilometers. And what is that? How does that feel when we look at something like the UK? Dark squares, 27 specimens per kilometre squared. Complete order of magnitude different. And you can see some of the areas, Borneo still under-collected, Sumatra under-collected, both of these highlighted in the report as needing more work, many new species still to be described there. Singapore, where I'm going, probably won't need many more collections, as you can see. And Java as well has been well collected over the years. And this is a little bit out of date, because a slide from last year, I'm sorry, but you can see we're still assessing species, and many new species are critically endangered or threatened before they're even described, and that's highlighted in the report as well. And that is something that we all need to do when we're describing species, is to engage with our red list community and really understand threats to new species. So in our New Guinea paper, we put forward 6 recommendations, and I think these themes come through the report as well quite well, and I think they'll be suitable for all of us. Let's train the next generation of resident plant taxonomists. Resident, that's a really important word. People in country. Digitize and unify historical collections. The resident plant taxonomists need to be able to see what we have here. We need to support taxonomy long term. It takes a lot of time and effort to write floras, to do expeditions. Expeditions are not cheap. You've got to fund them. You've got to process the specimens, curate the specimens, digitize them. So long-term financial institutional support. Plant field guides increase the number so that there's more non-specialists who can go out and identify plants. So I've published Trees of New Guinea myself, and Palms of New Guinea is currently being very close to publication, prepared by Bill Baker and collaborators. Unfortunately, mine's 2 kilograms, so it's not really a field guide, but it is available as a PDF if anybody wants it. Yeah. And support new international collaborations. Those resident plant taxonomists, they really want to plug in, they really want to take part in these initiatives like today. And of course, let's go and fill in the dark spots or the white spots, whichever colour spot you really want. And so just on an upbeat note, here we're trying to do some of these things. So this was us a couple of weeks ago. We did a tropical plant ID course out in Manokwari in West Papua, and we had the biggest poster ever made that I've ever seen. So driving past that, was a bit of a shock when we saw that. And these are a group of young university students, NGOs. They're going out, they're doing field work. They want to know their plants. They want field guides, they want taxonomy, and they want the latest names. They want the world checklist. They want to know the latest families, the latest genera, and the latest phylogeny. And this is the trees of New Guinea. If anybody wants a copy, just ask me, and a PDF available on request. And with that, I'd like to say thank you very much.
Brilliant. Thank you very much, Tim. That was absolutely fascinating, and thanks so much for keeping to time as well. So that means we do have quite a bit of time for questions, so I'd like to take questions from the floor. Okay, I'll start you off, Tim, as people are shy. So really fascinating about this idea of biodiversity hotspots versus wilderness areas, and how do we prioritize which to put our resources into, given that they're both important, and in some ways the wilderness areas are those that we could prevent biodiversity crises within, whereas of course biodiversity hotspots are ones where we already face a crisis and have urgency. So how do you balance those two?
Yeah, well, the hotspots, when the hotspots paper came out and New Guinea wasn't there, I had to readjust mentally, because we all use the word hotspot for so many things, don't we? The word hotspot is just like, it's a knife crime hotspot or a traffic hotspot. Biodiversity hotspot sounds great. Sounds like there's loads of biodiversity there. But actually, you have to use that. It's a very loaded term in our community now. And what happened with biodiversity hotspots, I was very disappointed that New Guinea is. I'm very happy that New Guinea wasn't a hotspot, because that means it's not threatened, but I was very disappointed because CEPF won't give any money to non-hotspots. So every time CEPF came out with these, and you've had CEPF funding, haven't you, quite a lot, and every time they came out with new grant calls, they're like, yeah, money, and oh no, I can't apply for that. So everybody thinks that wilderness is, it just got ignored, and it's just like, oh, they're fine. And so I think if we can highlight them somewhere, in another way, and I think, this dark spot idea, maybe people actually take notice and go, oh, okay, these areas need work. They need attention. So changing the narrative from that hotspots wilderness is needed, which will hopefully influence some funding. Because a lot of our stuff is all project funded, and to get really, really good chunks of money to do expeditions, people, red listing for somewhere like New Guinea, it's not cheap. Yeah.
Great, thanks, Tim. I think we've got a question online. Oh, in fact, yeah, let's take one from the audience first, and then we'll go online.
Okay. Thank you. So my name's Mike. Is this on properly? You can hear? Yeah, my name's Michael Aberton. I'm from the International Institute of Tropical Agriculture in Nigeria. So that's part of the CGIAR system. So it deals a lot with ex situ, of course, gene banks and so forth. But my question is really about this issue of taxonomy, taxonomists. Am I right to think that this is a sort of worldwide shortage of trained taxonomists, which affects both this work, affects our work in gene banks, and really, are there collaborations, are there more that we can be doing in terms of trying to address that?
So the question was, is there a shortage of taxonomists? Yeah, I think there is, but I think we now have to look at not becoming a two-flow system, two-stream kind of thing, where we've got all of us, like in the north, doing DNA, herbarium, phylogenomics, and then people, for example, in New Guinea, Papua New Guinea, who really don't have those tools at hand, and they still want a herbarium where the electricity isn't cut off and the air conditioning runs all day. So at the end of our course, we did a closing ceremony, and halfway through, the electricity went out, and we're just sitting in a black room. And so the fundamentals of taxonomy that we take, for example, for granted, such as a functioning herbarium, that they are not there. So to provide the institutions, the herbaria, which are cared for, with the people inside working on them. It's a two-pronged system, and that's a real shortage. So the whole of Indonesia, less than 11 herbaria, and no real functioning ones on Indonesia and New Guinea at the moment. So you don't have the facilities to actually do taxonomy as well. Yeah, and therefore the university students, they go off and do something else. Yeah.
Thank you. I think we've just got time for one quick question from online. So, Flo.
What alternatives do you think are viable for boosting plant research in countries considered underdeveloped or developing?
Oh yeah, that's kind of similar. I think David Attenborough always says something like, people are born loving the natural world. And when you go to somewhere like New Guinea or Papua New Guinea, there's all these students doing biology who absolutely love plants, and they love going in the forest, and they love all their bird-winged butterflies, and they want to do stuff. And then they go and see the herbarium and the forest lab, and it doesn't quite all work. So really helping to invest in those fundamentals so that people can do what they love I think is critical. Yeah.
Great, thank you very much, Tim. So I think we'll draw that to a close there. So let's round of applause for Tim. Thank you. Excellent, thanks. Okay, so moving on. A warm welcome now to Dr. Janaína Gomes-da-Silva, who's a botanical researcher at Rio de Janeiro Botanic Garden, and has broad interests in phylogenetic systematics of angiosperms, cladistic methodologies, historical biogeography, and linking all of that to the themes of conservation. And she's talking today on research and conservation priorities for endemic and undescribed Brazilian angiosperms. So, welcome. Thank you.
Hi everyone, it's an honor to be here and discuss about two topics I have dedicated my last 4 or 5 years to investigate it. First, I'm gonna talk to Brazilian angiosperms areas of endemism, and then I'm gonna talk about undescribed Brazilian angiosperms. The exceptional biodiversity of neotropical region has fascinated and intrigued naturalists, botanists, and biogeographers. And recent data indicated that neotropical region has greater biodiversity than Asia and Africa combined. Brazil, the largest country in South America, has high levels of biodiversity and endemism, and the Brazilian territory encompassed most of Amazonia and the 2 biodiversity hotspots, the Atlantic forest and the Cerrado. Describing this mega diverse flora is a significant challenge. Thus, the beginning was the extensive travel from Martians across the Brazilian territory and finished with Flora Brasiliense. And now is new phase over the last 12 years, the power of the scientific collaboration along with the mutual goal of reaching the global strategy plant conservations was the driving force responsible for unity a legion of taxonomists to carry out 6 projects to provide an accurate estimate of the number of species in Brazil. The project Flora in Reflora opened several or many opportunities for biogeographical and ecological research. Several studies have contributed to development of data of patterns and processes that shaped the current distribution of angiosperm in Brazil. But to date, no endemicity analysis has been conducted, including all endemic angiosperms. The areas of endemism são basic units of historical biogeography and should be a prioritizing factor in conservation programs. But where these areas for angiosperms? This study has two questions. What are the areas of endemism of Brazilian angiosperms, and is there a special mapping between the areas of endemism and the limited protected areas? In a short, this study was based for flora and reflora databases for all endemic angiosperms from Brazil. They study published the data paper and this study has extensive support information. The areas of endemism were identified for basal angiosperms, monocots, and eudicots using 3 grid size and 2 grid size, and combined criteria was used. Spatial congruence of areas of endemism, the area con highest score of endemicity, and the highest score of endemicity for area and for taxa. For basal angiosperms, the highest score is in Atlantic forest and Paraná forest. Here is the supporting for each area. For monocots, the highest score was found in Atlantic forest e Cerrado. And for eudicots, also the areas con highest score with different grid size is in Atlantic forest and Cerrado. The congruence of results enabled the identification of 5 best support areas of endemism and 2 generalized areas support for 120 angiosperm families. The results enable Cerrado is the unprotected area for Brazilian angiosperms. This is zoom, where you show the under-protected areas. The next step for angiosperms species is the time. This manuscript is under review. And now I talk about a little about future discovery and conservation priorities for Brazilian angiosperms. How many distinct life form species does your planet have? How many angiosperms are in your country? Described, undescribed, in what classification? This story illustrates how important is understand how many species there are on the planet. Currently, approximately 317 species of angiosperm are accepted, but studies indicate another 10% at least is still awaiting description. This number demonstrates that there is still a limited taxonomic acknowledgement of the world flora. To estimate the total number of species, including undescribed species, two questions must be answered. First, how many species described so far? Second is the question is more complex. It's required estimating the number of unknown species to science, or undescribed. Brazil has extraordinary biodiversity, but are Brazilian angiosperm approaching an asymptote? Which areas has the great potential for discovery? And in other words, what would be the impact on conservation priorities if all undescribed species were known in the catalog was complete? For answer these questions, we assembled full data sets from plant name index and edit manually. It's a very, very hard work. The method is too long, but in short, we use the number of species of discovered, description rate, and attributes, year of species descriptions, taxonomic effort, and taxonomic effort per interview. This is only esqueci a palavra. Passar. Human density and occurrence records for 4 million records were considered. In short, species level data, occurrence records, human density, fire, protected areas was combined, and statistical models and equations was used. So, where is the areas, where is the gaps for Brazilian angiosperms? The data indicated that Amazonia and Caatinga offers greatest opportunities for new species. Our findings suggest that around 19% of areas with high discovery potential do not overlap con protected areas. Some families with high discovery potential have seen a decline in species description. The Amazon and Caatinga and Pantanal, clear need for a national strategy for cataloging and funding for facility for collection and research, instructions for taxonomists working in the field. This graph must show the description rate for Amazon, but unfortunately, only half of the territory is undocumented. And many known species have a few geographic records. And 35 years, the deforestation of Amazonia has 4 times the size of the territory of the England. So, in short, the main gap is the time. Our data show that the deforestation scenario in Brazil is much faster than our capacity for species discovery and subsequent description. They highlighted the value of the conservation units in Brazil, which safeguard the regions which established biological significance. But unfortunately, the fire is there and the protected areas too. So, for conclude, conservation status is difficult or incomplete due to information gaps. In era of pervasive depreciation of taxonomy, escalating deforestation, and declining in plant collection activities, our data underscores the urgency for a substantial increase in future sampling efforts. The unknown species are concentrated in rare locales where there are high levels of habitat destructions. These findings highlighted the urgency of hindkind enthusiasm for additional exploration and taxonomy and increased resources for comprehensive scientific research. In Brazil, protected areas could play a role in salvaguarding many angiosperm species from extinction. Our results show that coverage of the angiosperm distribution by protected areas is not enough for effective protection with the current arrangement of Brazilian federal protected areas. There is a need for expand the coverage of protected areas to ensure the effective protection of species. The Union for Effective Conservation, the union of science, research, strong collaboration, society, and government and education is needed. Finally, for the planet, to combat the climate change and its impact, to protect the biodiversity and to protect each species, it's imperative to adopt this perspective. Thank you.
Very much. Okay, thank you, Janaína. That was absolutely fascinating talk. I'm sure there are questions arising. Yes, please, at the back.
I just wanted to ask you, I've travelled to Brazil, and my name is Penelope Dawson-Brown. I travelled extensively, and I remember the Pantanal as being the most beautiful place. absolutely stunning. And then recently I saw a program which showed that it was virtually, the habitat had been tremendously destroyed. And I wondered if, a lot of those plants had been recorded, and is it too late to record them because of the climatic change?
I don't understand the question.
Could you just repeat a summary of?
Sorry, I was asking you particularly about the Pantanal area of Brazil, which I visited about 15 years ago. Very, very beautiful. And then recently saw a program showing that so much of it had been destroyed through climate change, flooding. And I wondered how many plants had been recorded, and would it be too late to record them now? Because some might have been lost. Sorry, does that not make sense?
For the Pantanal.
Como está? Ah, okay. The question is excellent, but the outside the focus of this work and
Hi.
Hi.
Hi, Janaína. You talked about the time to actually assess all these species, but I was wondering about resources as well, because we see a lot of resources, especially international resources, that go into the Amazonian forest, but areas such as the Caatinga, they don't have as much appeal to conservation strategies, even though they have easy access for scientists to access that biodiversity. So I was wondering actually if the attention and the politics and also the resources that go into this research and also these strategies, they don't play a more central role towards targeting the type of biodiversity that we are prioritizing in conservation strategies at the moment.
Okay. The highest opportunities for undescribed species is in Caatinga, and the Caatinga need conservation. I don't. It's okay.
Yeah, that's fine. Thank you very much. I think we can just take one very quick question from online, please.
So biodiversity is disappearing fast, and we need to collect and study as much as we can. At the same time, institutions are required to publish as many as high-impact articles as possible. How would you recommend balancing these two contrasting requirements?
How I recommend?
Yeah, how to balance the need for fundamental discovery of biodiversity versus academic high impact, the need to publish in high impact studies in academia.
I don't understand.
Okay.
Okay. It's difficult, the question. I don't know.
Okay, that's something that we can, certainly that may become a theme, and we may just be able to explore that further tomorrow in the panel discussion as well. So thank you. Let's have another round of applause. Okay, so finally in this first session, we are delighted to have with us Professor Aída Vasco Palacios, a mycological researcher from the University of Antioquia in Colombia, with interests in tropical fungal diversity, ecology and conservation, and is presenting to us today on fungal diversity of Colombia. Thanks.
Okay, thank you so much for this invitation. For me, it's a pleasure to be in this auditorium after 15 years. I did an internship here, working in the fungarium, and now I am here as a speaker. So I am very happy and excited to be here. So I'm preparing this presentation with my colleagues, Viviana Motato and Tatiana San Juan. And today I am going to tell you a little bit about the state of the knowledge about the diversity of fungi in Colombia, the main threats, and the challenges. So just to start, okay. Colombia, I think it is important you know that there is in the north of South America. The Cordillera de los Andes is coming from the south and then split it in 3 branches. And they makes the country with a lot of landscapes and or a high orography that produce many types of landscapes. So we have the savannas of the Orinoquia in that area, the big Amazonian forest that is covered half of the country with a lot of type of ecosystems, the Andina area, the mountain region, that we can find like high-level paramos, oak forest, mixed forest, and also anthropic ecosystems. Then the beautiful Caribe with lowland tropical wet forest and dry forest as well, and the Chocó biogeographical area. That's made the country with a high biodiversity. We are now the 3rd country with most biodiversity in the world. The first is Brazil, the big ones. But, okay, we are like in some kind of competition, but Colombia is quite important as well. So we are the first in orchids and birds. But the important here is, what about fungi? What do we know about the Colombian fungi? And the history of the mycology in Colombia is only having 200 years. It's the same age of the country. We remember that we were a colony from the Spanish crown. And at the time of the Royal Botanical Expedition of New Granada, Colombia was getting her independence as well. So in that time, we have 4 periods. The 3 first, they are mainly by foreigner expeditions, and the collections are abroad. But during the last 22 years, most of the history of the mycology has done by Colombians. So we started with the Royal Botanical Expedition of New Granada, where mutis and collaborators were to steal the New Granada territory to documentary the flora and the fauna. Remember that at that time, fungi were plants. That's why they were just doing some draws. We found 14 species of fungi, 8 belongs to Ascomycota and 6 Basidiomycota. So it was the first time that the funga of Colombia was registered. But the first publication was from Humboldt and Von plant specimens that are here in the Kew Botanical Garden, so very important specimens that they were described by or studied by William Jackson Hooker, who described 73 species from Colombia, 12 of them new species from the world. I am not going to tell you about all the 200 expeditions or history because it's very interesting, but it's not the focus of this topic today. But something very important is in the last time, the mycology has been developed by foreigners, but in collaboration with people from the country. And this is very important. We are few, but we are trying to do our best. So currently, there are 96 research groups that work in fungal topics, not only in taxonomy and systematics or in ecology. Some of them are working more in bioprospecting and biotechnology, but at least they are focused in the Colombian fungi. In 2020, in a project that it was developed between Kew and the Humboldt, and also we are here as an association, the Colombian Mycology Association, we start to work together to know the state of the funga and the knowledge of the fungi in Colombia. This work was made by 40 authors, 31 of those that are from Colombia and from different institutions. It was an opportunity. We worked ad honorem in this work. At the end, we got this amazing book that you can download them from the internet, that is a catalog of fungi of Colombia. It was written down for the Colombian specialists in mycology in different topics. More than 32 mycologists, 27 of these from Colombia. And we are very proud of this work, and also because it's the basic line to know where to go since now. So what do we know about the Colombian funga? So at the time of the book that we collected the data, we found 7,079 species recorded for the country. These lists were checked by the specialists of each group. We did a currently review, and now at the 2023, we have 500 more species recorded for the country. So we are trying to work harder in this topic. Okay, some just data. Most of the species are from the Scomycota group, including micro and microfungi. The second more diverse group is Basidiomycota, including macrofungi and also yeast, environmental yeast, and other groups that are also represented. Almost 15% of this diversity are new species, and we have the idea that this number is higher. But most of these new records have not been described. They are still in their variants waiting for the specialists to go and study them. What does it mean, this number are in a global context? So here there is a map with some data about global species from countries. And here, look, the United Kingdom, too many specialists, a long history of research, and for now they have 18,000 species of fungi reported for the country. Also, Italian, it has 23,000. Our close competitor, Brazil, that they have, okay, we work together. But with the Brazilians, that they have been like, they have now too many specialists at university working in the Brazilian fungi. They have reported until now. 2015 is that date. So I think that it has changed a lot. It's almost 20,000 species. So we have a lot of things to do. Here, how many species do we expect? There are different approaches, but I am going to focus on these two. The first one is thanks to the hardware and looking approach that hardware is here, sitting in the front. It's a pleasure. So in the tropics, the range or the relation between fungi and plants range between 6:1 and 10:1. And in Colombia right now, there are 29,000 of plants. So this number came out from this approach. Also, if we use the approach that 10% of the whole biodiversity is held in Colombia, so we will have between 2,200 and 380,000 species. And we are waiting for a high diversity, based in this work published in 2014 about soil fungal diversity. We see that the Amazon in Colombia is one of those hotspots of biodiversity. And similar, when we are checking the global patterns in endemicity on vulnerable fungi in soils. So we are expecting Colombia to be a hotspot of biodiversity. The collection efforts and information gaps. In red, maybe it's not the best color for the high, the most of the collection has been done in the Andean region because it's close to the biggest cities and where is the main ways are located. But other regions like the Caribbean, Choco biogeographic, the savannas of the Orinoquia and the Amazon are almost or not well studied. Here, based in Basidiomycota and macrofungi data, on red we have departments, all regions, without any report of fungi. Look here, the Guaviare, and those ones in that color are only having from 1 to 5 reports. So most of the country is still not well known. Similar to other groups, okay. Most of the country is unexplored. Key ecosystems like dry forest, Amazonian, biogeographical, Choco area, or Antioquia are still little explored. Few resources for research. It's difficult to find money to do science in fungi, and few mycologists that have a permanent position in institutes that they can develop very well the science. Similar to other groups, like rose and smut fungi, I am not gonna stay here. Main threats, okay. With main threats, we are now working in fungal conservation in Colombia. In a first evaluation, we found that there is few legal instruments that can be related to the protection of fungi. Only some resolutions and regional agreements for protect lichens. And I think that because lichens are green, most of them, and they were confused with epiphyte plants, so they just included lichens in the legislation, but the other fungi are not protected. Plants and animals have lists of threatened species and that may be visible to guarantee their protection, but not fungi. By now, we have evaluated 25 species, but we hope that this number will increase in the time because we are working with several colleagues trying to improve the red listing task that we think that is very important. Sorry. What are the main threats? So, in general, the deforestation. Why the deforestation? Because we are demanding on fuel, that people are still using wood as fuel, energy networks, mining, cattle raising, industrial agriculture like the palm oil is a big problem in Colombia right now, and infrastructure projects. Also, we have detected another trait that there are the invasive species. Amanita muscaria is now invasive, the Quercus humboldti root tips. So now it's switching hosts. So we are starting to talk about invasive species also on fungi. As I told you, the deforestation, look where the focus of deforestation are, close to the Amazon, all this Amazon border, where we have empty the areas about information on fungi, of few records are in danger now. Also, these spots, and what about the other part of the country? This whitish color is not because it are not in danger. There is not forest anymore. So look, we have our native forests that are all with some problems in deforestation now. So too many challenges. What are we trying to do? Fill in the gaps with new projects. For example, I got this a small grant from a Spoon, and during this, when we went to the Guaviare department, this department that it was in red because they were not reported on fungi before, and we were with my colleagues, with Tatiana San Juan and with Viviana Motato, we were collecting on there. It has music, but not, not here. But okay, the important is that it's a transition area between the savannas, the Orinoquia, and they have some white sand forests that they origin from the Guyana Shield. So the diversity in that point is very, very high, but there is a big problem because deforestation. So, yeah. I am not sure this is the time. No. Okay, I am okay. Sorry. So we were collecting saprotroph, ectomycorrhizal fungi, and also entomopathogenic, because Tatiana San Juan is working in that now. And the project is more basic in soil diversity, but we were in the place, so we had to collect mushrooms as well. This is the white sand forest. Okay, I just go follow and okay. From this work, we collected more than 200 in the past one. And we collected more than 200 specimens, and we are expecting to have like 80, at least 80 species that they were at like 30 new reports to the country based in the previous list. Other project that we have developed is in the tropical dry forest. The tropical dry forest is very, very special forest in the country, and most of the 80% of this ecosystem do not exist anymore, have been deforested. So for this project, we have collected 101 species and 21 new reports to the country. So somehow, remember this map, we have filled up this empty space, and then with the Tolima, we hope that we will have more than 100 reports. So somehow with few steps, and now based in that we know where are the gaps, we are trying to fill it up the work. Something very important, and it was mentioned by my colleague before, is a strain national collection on fungi. We have two types of collection, the Fungaria collection and the strain collections. And now we are also trying to strengthen the strain collection with macrofungi, because those are very, very important to not only for taxonomical and distribution information, but also for the biotechnology. Our country is now focused in the bioeconomy, so we need to provide strains and data about how to use our biodiversity for the production of food and feed, pharmaceuticals, chemical products, and energy sources in order to develop our country in a sustainable way. And at the end, also, consolidate fungal conservation. Now we have the UCN Colombian Fungal Specialist Group that has been started in 2022 with the aims of promote the conservation and knowledge of fungi in Colombia. Also to generate information for stakeholders and decision makers, because it's a big hollow between the science and decision makers, and we need to fill it up to give them information that they can use for make policies. And we are starting also with the national red listing, that is also a very important tool. So that's all about the Colombian experience. So thank you so much. I would like to thank my colleagues and all of you to be here. Thank you.
Many thanks, Aída. That was absolutely fascinating. And I think all botanists in the room probably think that we sometimes think we've got challenges, but that sort of brings it home, the scale of challenge and that biologists can face. So do we have any questions, please? Yes, please, in the center.
And we need to
just
work
wait
with
for
the
the
botanists together, because most of the fungi are having symbiosis with plants. Very important.
Yeah.
Thanks a lot for the wonderful work you're doing, Aída, and the very enthusiastic lecture. I was wondering about, I'm Mieke Verbeken from Belgium. We never met in person, but hi. The 35 fossil fungi, can you explain? That's an impressive list. It was in the list that you have 35 fossil fungi.
35 what?
Fossil?
Fossils. Okay. Yeah, sorry. It's because in Colombia it was Thomas van der Hamen, a botanist who used to work with palynology. And they made many explorations in the country, try to understand the history of the vegetation. And they found some spores. and they describe them, so like fossils. So those data are in these palynogeological studies.
Spores in dung or
spores,
okay,
mainly
okay.
spores, not basidiomas or vertebrates.
And they are identified?
Yes, they have names. Yeah.
That's wonderful as well.
Yeah. in the platinum fungi site that is hosted in Kew, you can find the names and maybe the reference of that.
Thank you. Yes.
Okay.
Yes, over this side, please. Thank you.
Thank you very much. My name is Eric Bow. I've visited Colombia lots of times. Beautiful place, fantastic diversity. I wondered, and this is a question not just for fungi but for plants, what is the scope for tourists or paying visitors to help you with your work? Because I know that in Colombia, Dan Winkler occasionally has tours to go there. Do you think this is something that could be developed further to help you in collections?
Yeah, okay. I am not for helping collection, because to collect in Colombia you need a special permission, even though you are a foreigner. You have to collaborate with local research. But I know that David Winkler is working with Athena San Juan, one of my colleagues, and they are doing tourism around, like fungal tourism. And now this is an important tool also for local development, because the money of these tourists, these tourisms are also go to the community, and they help to preserve the forest and to know the importance of fungi. So now this is a new tool, the fungal tourism, like the birch washing, like fungal washing. I don't know what's the name of that, but yeah, it's important. It's important.
Thank you.
Thank you. We have a question online. Flo?
So what is the difference between macro and microfungi conservation, and what are the challenges with microfungi conservation, and why is this important?
This is a topic for a workshop in conservation. Okay. It's different because the ecology and the distribution of the different type of fungi is very different. And also because with the macrofungi it's more easily to see the distribution, but with the microfungi, the tools that you need to know the real distribution and to know the threats and to release them are quite difficult. But now with the conservation group, we are start to thinking how to work in microfungi as well. Yeah.
Any further questions in the audience?
Hi, thank you. Karen Bacon from Ireland. That was really interesting. One thing that caught my attention was you mentioned invasive fungi, and I was wondering what's the problems that they cause? How much of a problem are they?
Okay, the main problem is that we don't know the effect of Amanita with the local fungal community. So we know that they have switched from pines to oak forest, to pine from Quercus, because it was detected from the root tips. So we don't know what is going on with the local and the native fungi. That's the point. So, okay.
Thank you. I was just going to say, I think one of the things that comes through from your talk and from experience in other countries is the importance of having champions like you that will do a lot and try and make these bridges between the scientists and the regulators and so on. I think this is a very important point. And I think if focal people can be identified, they can be supported. This is a very important step forward. Thank you.
Thank you, Dave. Yeah, we are trying to do our best. And I think that something that has happened very important in Colombia now is the creation of the Colombian Mycologists Association, because now we are a group of people with different expertise, and we are working together trying to do many things, like to visualize fungi, to work with policies, to work with education. We cannot do all those things only one person. We need a team, and now we are a team, the Colombian Association of Mycology.
Excellent, thank you. Oh, Domitilla.
Yeah, thank you. I just want to congratulate you on a fantastic talk, and also to congratulate Colombia on forming, I think, the first regional fungal specialist group for the IUCN. I don't know of another country that has one, so congratulations. I mean, amazing, really.
Brazil is having one.
Brazil also has one. Okay, I didn't know that.
But well, we were the first.
Okay,
we were the first.
We've got to watch the competition here between Brazil and
No, it's not
Colombia.
a competition. We work together. Yeah.
I'm just wondering if there are, any of the, you talked about the different groups, but in terms of the red listing, if there would be one where you feel that the taxonomy is maybe good enough to get a picture of what might be going on for the group in the country. And globally they're suggesting that we do lichens and try to do lichens in different countries, but I was just wondering if you'd thought as the group yet, which ones? Are you going to just do a few that you know might be threatened, or are you going to try to do a group that might tell a story of what's going on with fungi in Colombia?
Yeah, I think that depends on the founds. Because we really want to do like a big expeditions covering all the groups, but we don't have money for that. This small expedition, it was just a project for soil fungi, and I took my card, took my colleagues, and said, okay, I will pay for everything, but we were in a cheaper hotels and everything, because if there is not another way. But yeah, we were trying to see if maybe with the government or with some external support, we can try to do a big expedition in Colombia to fill up all these gaps, but also with all the groups, because we have lichenologists. So there is a big group of lichenologists in Colombia that the leader is Viviana Moncada, and we have specialists in entomopathogenic, saprotrophic, polypores, corticoid, ectomycorrhizal fungi, also from dipterocarps, that we have dipterocarps in Colombia as well. So we have the people. We don't have now the resources to do more, at least at that point. So,
Thank you. Okay, so I think let's say
Thank
that
you so much.
brings to a close the first session today, but the theme of where are the diversity hotspots and critical knowledge gaps will continue tomorrow in session 2, and all our speakers of today will also be on the panel tomorrow, so there'll be more opportunities to ask questions then. So please join me again in thanking our speakers today and their illuminating talks. And so I have one announcement to make for people who are online. So the next online session will be the flash poster presentations at 5:30 this evening, so please join us then. Otherwise, I think now we'll stop the recording.
Please have a seat. Please take a
will get my timer ready and introduce the first speaker, who is Dilzara.
Good afternoon, ladies and gentlemen. Do you hear me? I am from Azerbaijan, working at the Institute of Botany, Ministry of Science and Education of Azerbaijan. I have to say that Azerbaijan, as a part of the Caucasus, is biologically rich and endangered hotspot of the world. A view of climatic and geographic diversity, the territory of the country was divided into 20 botanical geographical areas. First assessment of the plant was performed in 1989, and 130 plant species was included to the Red Book, first edition of the Red Book, and second edition was much later in 2013. And about 300 fungi and plant species was included into the second edition. This year, just recently, third edition was published, and 460 plant and fungi species, mainly macrofungi, was included to this one.
Fantastic. Thank you very much indeed. Okay, next up from Oman Botanic Garden, we have Lila.
Well, actually, this is Abdurrahman. So
oh, okay.
I am presenting on behalf of my colleague Layla Al-Harthi. So I'm Abdurrahman Al-Hinai. I'm a senior botanist at Oman Botanic Garden. Oman Botanic Garden is a newly constructed garden in Oman with the aims of exploring, conserving, collecting the biodiversity of the flora of Oman. So in the garden, actually, we are actually collecting we are managed the team actually able to collect and conserve actually about, well, I'm a bit confused. We conserve about more than 60% of the flora of Oman. We manage actually in the garden we collect
50 seconds.
Okay.
Not very long, is it?
Yeah. Okay, sorry. So that's panic.
Okay, thank you very much. Okay, so next up we have, talking on evolution of pollen and Adesmia clade, we have Higor. Welcome, Higor.
Thank you.
You ready to go? Off you go.
Hi, everyone. For some of people, pollen grain is just a yellow ball or a black and gray balls or using to the pollination, but for me, for us scientists, pollen grains is a complex, a group of characters and traits that we can use for the taxonomy and to the ecology and the paleobotany and this kind of thing. So to Adesmia clade, for example, the pollen grains can vary in number of aperture, the morphology of the aperture, the ultrastructure, and the ultrastructure of the extant layers. So to this clade, we cover at least 20 traits, and we can recover for each clade at least one plasomorphic or one synapomorphy or one homoplasy to this clade. So with this, we can maybe use this data to a new classification, like a subtribal classification to Adesmia clade.
Thank you very much.
You.
And next up, we have Wahid.
Yes, hello, thank you. I'm Wahid, and I'm a botanical scientist at Species 360, where we are working with information at a global level serving conservation. And my talk poster is all about filling in the gaps. Now, we see from the State of the World's Plants and Fungi Report 2020 that 2 in 5 plants are estimated to be threatened with extinction. But species extinction can be prevented, and the global community of Botanic Gardens is our best hope. However, 2/3 of ex situ plant diversity in Botanic Gardens is unknown, and 80% of the world's Botanic Gardens do not have good quality or comprehensive plant records. So without good quality data, how can we do plant conservation?
50 seconds.
And that's something we are addressing in terms of complex usability and ownership in terms of software, and we hope to do that with our product called Hortis. So if you want to learn more and learn about plant records in Botanic Gardens, find out how we're rethinking all of that.
Fantastic. Thank you very much indeed, Wahid. And next up, we have Eileen. Eileen, welcome.
Hi, everybody. So I'm talking about a project that I have at Natural England, working in conjunction with UKCEH, their Centre for Ecology and Hydrology. And it's a project here in the UK looking at the distribution of grassland fungi. So here in the UK, we've got a particular stronghold for grassland fungi. It's an internationally rare habitat. And we have over 85,000 records, which is an amazing number. But even with that number, we have real gaps in that data. And so we're working with some modelers at UKCEH to develop a species distribution model that we can then use as a tool for people on the ground who don't necessarily have expertise in mycology that they can use so that they don't accidentally threaten these important grassland fungal habitats by things like woodland creation and other types of environmental change.
Fantastic. Look at that. Saved us 15 seconds. Well done, Eileen. Next up, we have Lauren Baker.
Hi, my name is Dr. Lauren Baker, and I'm the Secondary Education Officer at the University of Oxford Botanic Garden and Arboretum. We have a broad portfolio of collections-based research at the garden, which informs our education and public engagement programmes. Recent work that we published in Plants, People, Planet, led by Dr. Chris Thorogood with a team of foresters and conservationists in Southeast Asia, examined the extinction risk of Rafflesia, the world's largest flowers. Chris also was involved in the Philippines' first ex situ propagation experiment this year. Other research is interdisciplinary. For example, Chris is working with mathematicians using modelling and has showed that different Nepenthes pitcher forms capture different prey, and how and why giant Amazonian water lily leaves grow so large. The latest project could inspire offshore floating solar farms. Our final research project is in collaboration with the Department of Psychiatry in Oxford, and it is investigating green spaces as non-clinical labs for researching brain health in response to a dose of nature.
Perfect timing. Well done. Thank you. Next up, we have Ludwig. Off you go, Ludwig. Your minute starts now.
Hello, everyone. I'm Ludwig from the Royal Botanic Garden, Edinburgh, where I'm a PhD student. And as we have heard throughout today, our knowledge of plant biodiversity remains incomplete. And this is particularly true when it comes to plant biodiversity occurrence data, which is still severely distorted and makes us unable to set conservation priorities at a fine scale. So in my work, I am going to tell you how many species you kind of need to reliably recreate biodiversity patterns based on the World Checklist of Vascular Plants, and how you can use that information to assess bias in conservation data sets. So find me at my poster if you want to know more. Thank you.
Fantastic. Thank you, Ludwig. And next up we have Matilda.
Anecdotally, we know that more and more of the 2,000 plant species we're describing each year are already threatened with extinction, but we've never been able to put a number on it until now. We looked at the World Checklist and the Red List and looked at the proportion of species described each year that are considered threatened. And we found this really striking statistic that of the species described in or after 2020, 3 out of 4 of those species are already threatened with extinction. In light of this result, we're now calling for all newly described species to be threatened with extinction right from the point of description. And ideally, if you can assess them when you're describing them, by all means, please do so. Find me at poster 9 if you want to hear more.
Fantastic. Fantastic. Strong message too. Thank you very much, Matilda. And up next we have Iain A.
Yeah.
Oh, no we don't. Yes,
Okay.
Maria, sorry.
Yes.
Maria. Off you go,
So
Maria.
there's no easy way to say this, but we are losing a lot of diversity. So we need to take action to protect it. So what happens when you need to protect the most diverse desert in North America? Which one of the more than 4,000 species should be prioritized? And most importantly, where can we find them to preserve them in ex situ collections? So basically, those were the questions that we answered with this project. First of all, we created a list of species that we can prioritize, and also we identify the hotspots to collect those species in the future. So This information will lead the way to increase the representativeness of the Sonoran desert flora in seed banks. And yeah, we all want to protect this amazing biodiversity. So if you want to know more, find me in poster number 10.
Excellent. Thank you very much, Maria. And we're making good time here, actually. So next up we have Yan Wen-chen from University of Hong Kong.
Hello, everyone. I'm Iain Wen-chen from University of Hong Kong, presenting the poster number 11. Our research provides evidence for fungus-mediated plant pollinator mutualism in Monuloii, in which the flowers produce stigmatic and petal nectar as nutritive rewards and petals as bird sites for pollinators. The fungi are inoculated onto the flowers by foreign visitors and environmental and air dispersal, which gradually multiplied on petals under moist weather. The fungal hive serve as a food source for the insect larvae after petal abscission. The larvae develop into adults within 3 weeks and are then able to pollinate other flowers of the same species. Petal nectar can be regarded as an evolutionary adaptation to raise pollinators, enabling the flowers to benefit from increased pollination activities. Yeah.
Very good timing. Thank you, Yan Wen. Unfortunately, Natalia from Royal Botanic Gardens, Edinburgh, isn't able to make it, so we go straight on to Joette. Oh. Ah, okay. So. Ah, we have a little bit of chaos going on. That's what the hour is famous for. So you'll be coming at the end, but for now, we've got Joette coming. Thank you.
Yes, hi. I'm developing methods to make the reintroduction of wood-inhabiting fungi more successful and cost-effective enough to be implemented in real-scaled restoration works. So this is a complementary approach to habitat conservation to give locally disappearing species a jump start in protected forests where long distances and recent landscape fragmentation might prevent their natural return. I've been improving cultivation techniques from the lab stage to spawn production, and we have inoculated hundreds of logs to translocate throughout Finland and Sweden. While this research is really early, I'm hopeful that it can be adapted and implemented globally where it is practically and ethically appropriate.
Perfect timing. Thank you very much indeed, Joette. And up next from University of Coimbra, Portugal, we have Susana Cunha.
Hi, I'm Susana, and in this work we analyzed the red list assessments of 94 fungal species, which are in monotypic lineages. Our goal was to help inform fungal evolutionarily distinct and globally endangered, or EDGE lists for fungi, and our preliminary results showed that the vast majority of species in this data set was assessed as data deficient, which is shown in gray, and this really hinders the compilation of an EDGE list. We also can see that in most cases there were no molecular data or phylogenetic data, good sound phylogenetic data, which is seen in green as the basis for their monotypic assignment. So we urgently need to increase taxonomic knowledge and increase search efforts specifically for this species if we want an EDGE list. And you can find this at poster 14. Thank you.
Thank you very much, Susana. And up next we have Rafael from Royal Botanic Gardens, Kew.
Hi, I'm Rafael Almeida, and we're making a very interesting photograph field guide for common grasses of Madagascar. This book's gonna be bilingual, both Malagasy and English language, comprising about 300 species and covering at least 126 genera of Malagasy grasses. And we're doing it in partnership with KMCC and other institutions in Madagascar. And we're going to have, each species is going to have a specific page with photographic information from field work and information on morphological, ecological traits, and distribution, common uses, and common names. We're hoping that this book's gonna be like a foundational resource for local farmers in Madagascar, and it's gonna help improve cattle and other economic things related to the Malagasy grassy biome. Thank you.
Perfect timing. Thank you very much, Rafael. And up next we have Anne from Cambridge.
Hi, I'm Bernes, doing a postdoc at the Cambridge University Herbarium. And we are talking about species a lot during this symposium, and what people don't always know is that species concepts are actually tied to a physical specimen of plant or fungus that is deposited in a herbarium or a fungarium. And these are called type specimens. And these type specimens serve as the reference for the scientific name. So it's really, really important for taxonomists to have access to these specimens so that they can use them to build solid species concepts, identification tools, these kind of things. And my job is to try and find and identify the type specimens in the Cambridge herbarium so that we can digitize them, upload them, make them more accessible. And Cambridge has 1.1 million specimens, and they haven't been catalogued, and they're from all over the world for about 300 years collections. So looking for types in an uncatalogued collection of this size is a bit like a treasure hunt. And if you want to know how I do it, you can come and have a look at my poster.
Again, perfect timing. Thank you very much indeed. And up next we have Felix from Kew Gardens.
Thank you. So the edge metric is a metric that measures how unique and threatened a species is. And this has been applied to tetrapod animals, including mammals and birds, but to only one plant group so far, the gymnosperms. So to apply this to angiosperms is quite difficult because of the sheer size of the group, but also because of the limited coverage in phylogenetic and extinction risk data there is for angiosperm. So in order to do this, we use an imputation method that adds missing species to a DNA-based backbone tree, and machine learning to complement red list assessment extinction risk. And with this, we produced the first edge list for angiosperms. So we found that there was 28,000 species of angiosperm that qualifies as edge species, and that about 20% of all the angiosperm evolutionary history is at risk of extinction. And we hope that those edge lists will be able to put plants on an equal footing with tetrapod animals.
Perfect. Thank you, Felix. Excellent.
Thank you.
Up next, Tilahan.
Okay. This is Tilahan from University of Gondar, Ethiopia. As you see, this is rot is a primary rice disease in Ethiopia with 100% incidence and 44% severity, but the causative agent associated with this is rot has not been reported in Ethiopia. Hence, this study was intended to identify sheath rot causing fungal isolate using morphological and molecular tools, and to find out the degree of pathogenicity between those isolates. The outcome of this study will provide a platform for rice research and will pave a foundation for rice crop improvement to ensure food security in Ethiopia. More importantly, thus finding contributes to better understanding of the diversity, taxonomy, and pathogenicity of the identified fungal species for the development of effective management strategy. For detailed information, see you soon on my poster. Thank you.
Great timing again. Thank you very much indeed, Tolliver. And up next we have Ellie Graves from Royal Botanic Gardens, Kew. Off you go, Ellie.
Hi, I'm Ellie Graves. I'm the Digital Collections New Acquisitions Manager here at Kew. And my job and my poster is about developing workflows to deal with our new acquisition specimens to Kew Herbarium. So we get about 15 to 20,000 new acquisition specimens every year. And so we are developing workflows that take into account requirements for museum accreditation, taking considerations about our large-scale digitization projects happening currently, as well as looking at our current protocols and seeing how they can fit together. So we've now established workflows to deal with the majority of our new acquisitions, but the next step is dealing with material arriving with digital data. So that might be specimens arriving with digital data that was collected within the field, but also collaborating with other institutions to send digital data along with our physical specimens, so we can encourage some knowledge sharing between institutions and decrease duplication of effort. So yeah, I'm at 19, yes.
Perfect. Perfect timing again. It's amazing how much information you can cram into a minute. All of these talks just under a minute. Perfect timing. So up next we have Laura Kaur, I think. Yes, sorry, we've got a couple of dropouts. So off you go, Laura. Thank you.
Hi, everyone. We've heard a lot today about the threats facing plants and fungi. And to help tackle this, the Important Plant Areas approach was launched in 2002, aiming to identify and protect the best sites for plant conservation around the world. Over the last 20 years, IPAs have become the leading spatial conservation approach for plants and fungi. However, a global review was lacking. So working with colleagues in Plant Life and Kew, we've been asking where and how have IPAs been identified? When has this led to conservation action? And how are IPAs perceived? By reviewing hundreds of documents and interviewing 47 experts, we've brought together perspectives from nearly 80 different countries. Generally, there's a positive perception of IPAs, but this really varied between location and the different aspects of effectiveness. So please come have a chat with me at poster number 22 to learn more about our recommendations for area-based conservation approaches for plants. Thank you.
Perfect timing again. Thank you very much. Just working. Oh, so up next we have Georgina from Flora and Fauna. Off you go, Georgina.
Hello. Fauna and Flora is working on the in situ conservation of 3 threatened pear species in Kyrgyzstan and Tajikistan alongside local partners. So the key threats to the species are livestock grazing, over-exploitation of the wood and the fruit, and they're also not reproducing well in the wild. So we're working with local communities, the Forest Service and other local stakeholders, increasing their knowledge and skills to look after these trees themselves. Key conservation interventions include fencing and other grazing management measures, engaging communities in participatory forest management, supporting sustainable livelihoods, and we're also planting nursery-grown seedlings to boost the populations of these trees. Monitoring the project is providing evidence that local knowledge and behaviours are changing and that there are positive trends for the tree species as well. We're grateful to Fondation Franklin for supporting this work.
Thank you very much. Very much. Okay, up next we have, I've lost my place. Jose, Jose's not, it's Myra.
Yes, hi. Well, Bolivia is mega diversity country for more with 16,000 species for plant vascular. Is the first Latin American country adopt the program TIPAS. Today is identified 37 sites in the 2 species rate ecoregion. More than 300 species meet for TIPAS and criterion UCN evaluation. In the process, we have identified 6 centers of center diversity of useful plants and evaluate the risk of habitat collapse following UCN criterion, and discovered 12 species, new species of the science, and training 15 Bolivian professionals and 16 students. 10 worth of TIPA sites has been adopted for departmental government in Santa Cruz for conservation planning, and recent is also helping in the review of the key biodiversity areas of Bolivia. Please come on and read my poster is 25.
Thank you very much. And up next we have Ethan from Edge Hill University.
Hi everyone, I'm an undergrad from Edge Hill, and my project is all about roundabouts. Unfortunately, my lovely photo of a roundabout hasn't worked, but what it does demonstrate is environments. They're very dynamic, right? So when we hear the word roundabout, we're probably thinking like poas and like lowly and perennial. Well, actually, that is a roundabout, and it had a wet meadow on it. We found some orchid hybrids and stuff. The project is sort of, I call it a soil-up approach, because we've got soil environment, microbes, plants, but you can come and hear about it. I think I'm poster 26. Thank you.
Thank you very much. We'll all come and have a look at your roundabout. Thank you very much. So up next we have Sandra.
Hello everyone. Yes, I'm a PhD student from Galway, and my project is part of a larger project which deals with extinction in the fossil record. And basically we want to investigate on the issue of, well, we don't have evidence for plant mass extinction in the fossil record, but on the other hand, we have roughly 45% of the world plant nowadays endangered. So that leaves us with 2 options. Either the fossil record is not showing us what we would expect it to show, or the second option would be that what's going on right now is actually worse than everything before in the geologic history. So my part of the project is dealing with decomposition. So I'm looking into decomposition rates, and I want to identify relationships between certain leaf traits and environmental factors that will tie into the probability for a species to get preserved within the fossil record or not.
Thank you.
Yeah,
that's your minute. Thank you very much indeed. And up next we have, just to let you know, if you were wondering, and I'm not suggesting this is an ordeal for you, but we are halfway. So up next we have Lydia.
Soil water repellency occurs when hydrophobic organic compounds accumulate at the soil surface after long drought periods, and this can increase soil erosion. The role and biodiversity of fungi is still unexplored in these soils. So, in our study, we wanted to study the biodiversity of soil fungal communities in 2 sites with extremely water repellent soils in a valley in Switzerland. We discovered 2 fungal communities that are quite restricted, only 200 taxa. And this is the first step towards future studies on the role of fungi in soil water repellency and its complicated management. If you're interested in more details, please come to poster number 29.
Excellent, fantastic, well done. Thank you, and very good timing too. And up next we have Precious from IITA.
Hello everyone, my name is Precious. To battle hunger and ensure food security, there is need to explore climate-resilient crops and make them available. African yam bean happens to be a climate-resilient crop. It produces both tuber and seed, and it fixes nitrogen into the soil. The challenges we are facing for African yam bean is inter and intra variation. The tuber production is not consistent because you can have tuber in rep 1, you won't have tuber in rep 2, you will have in rep 3 across location, just like that. And African yam bean is proteinous. The tuber contains 45% carbohydrates. For more information, my poster is number 30.
Perfect timing. Thank you very much, Precious. And up next we have Harrison from University of Galway.
Hello, I'm Harrison O'Rourke, a PhD student studying the relationship between leaf traits and extinction risk in both a modern and a paleo context. So it's widely believed that plants don't suffer mass extinction on the same scale as animals due to a lack of evidence across previous mass extinction boundaries. However, plant groups at greatest risk of extinction may not be preserved in the fossil record, and this influences our view of extinction in the fossil record of plants. So if certain leaf traits of species are associated with extinction in modern floras are also associated with extinction in the fossil record, then it's the preservability of these leaf traits which influences our view of plant extinction in the fossil record. Thank you.
Thank you very much indeed. You're doing a great job of keeping to time here. Thank you very much, speakers. Next up, we have our very own Sebastian Pippins.
Yeah. So hi, everyone. My name is Seb. I'm a second-year PhD student based here at Kew, supervised by Felix Forrest. And my research focuses on edge species, those that are evolutionarily distinct and globally endangered. Think something like a Venus flytrap. More recently, I've been looking into areas of threatened evolutionary history. And what I've found is that when you compare the priority regions of 6 different taxonomic groups of both plants and animals, there are very few areas of total shared priority seen in red on the big map. But there are lots of areas of unique concern, such as Turkey for flowering plants, Iran for reptiles, New Zealand South Island for birds, and so on. Of course, this has implications when it comes to conserving the tree of life. And if you want to chat more about this, I'll be by poster 32 with the big Venus flytrap.
Fantastic. Thank you very much indeed, Seb. And up next we have Daniel from the University of Antananarivo. Welcome, Daniel.
Use of mycorrhiza to improve restoration success in tropical forests southeastern Madagascar. Madagascar is one of hotspots of biodiversity, and in its southeastern part there is a corridor called COFAV, in which is this home of 5 endemic families and a priority site for conservation. We aim to reconnect and to restore the remaining fragments by inoculating seedlings, and the results show that the use of native arbuscular mycorrhiza and ectomycorrhiza improve seedling survival rate and growth. For more information, poster number 33.
Thank you very much indeed, Daniel. Again, excellent timing. And then up next from the Kew Madagascar Conservation Centre, we have Landy. Off you go, Landy.
Hi everyone. Orchids are a high-profile group in Madagascar with near 1,000 accepted species, of which over 85% are endemic. This family represents, therefore, an excellent model for studying diversification and historical biogeography. For the project comprises 4 chapters: a comprehensive phylogenetic framework of Madagascar orchids at generic level, diversification and historical biogeography of the Colabi tribe in the Calanfio Alliance, conservation status of Madagascar orchids, and innovative species identification of Madagascar orchids from digitized images and iNaturalist website. For further information, please visit my poster number 34. Thank you.
Thank you very much, Landy. And then up next, also from the University of Antananarivo, we have Fitiavan.
Hi. In Madagascar, there are 24 genera of Cyperaceae. We choose to focus on Barbostalis because species on this genus, commonly called hersages, are one of the few multi-species endemic lineage on the island. Barbostalis represent the second largest endemic radiation and the only large radiation in a lineage using C4 photosynthesis. Limited DNA data are available for the group, resulting in a lack of an infrageneric classification of the genus. And lack of DNA data hampers studying character evolution, ecology, and biogeography of Barbostalis. Also, without a detailed DNA-based study, the limitation of species is uncertain, so our goal is to gain a clear understanding of the diversity and evolution of Barbostalis in Madagascar. Thank you.
Thank you very much indeed. And up next from the University of Sheffield, we have Hannah Ronan-Brown.
Hello, I'm Hannah. So my poster has a very long title, for which I apologise. I've tried to summarise it in a question, and that is, does the degree of pollinator reliance constrain plant defence against herbivory? So we can test this in wild tomatoes, because we have selfing and outcrossing wild tomato species. So we took several species that self and several that outcross, and we damaged them. And we measured their induced response. We also measured their constitutive defence levels. And we found, as you would expect, that leaves and flowers are differentially defended. They are different tissues, after all. Interestingly, we found that pollinator reliance does have an effect on the plant's ability to defend itself, which is an interesting line of research by integrating the studies with pollinators with studies with herbivores. So thank you. I'm at poster 36.
I'm definitely going to that poster later. And up next, I need to turn my page and introduce Rainey.
Hi, I'm Rainey with Fauna and Flora, and I'm going to present to you today about 2 of our Threatened Trees projects, which exemplify our context-driven approach, where we focus on communities as the long-term guardians of the land. Our first project is in Quang Ba, Vietnam, primarily on Magnolia grandis species. The primary threats there are cardamom cultivation and illegal logging. We've been able to successfully see 7 years of no evidence of logging due to patrols led by communities that we've established there, as well as community nurseries that have successfully transplanted more than 5,000 seedlings into the wild. And then also our second project is Raja Ampat in Indonesia. The primary species there are Raja Ampat palm. The primary threats are also logging. And if you want to learn more about it, I am at poster 37.
Great. Thank you very much. So this... okay, we were going to look at Anna's poster. Are we not going to look at Anna's poster? No, okay, that's fine. We'll go straight on to Maribel. Thank you very much, Maribel.
Hello. Today I'm going to briefly tell you about how genome size in flowering plants can play a role in influencing their extinction risk. What happened?
You had the wrong slide.
Oh, should I start again? Oh.
Oh, Anna couldn't be with us, I'm afraid. She had to go and collect her son. She just nipped off, and I did say that we would show a slide, but we're going to move on to save time. So over to you again.
Here we go. I got a trial run.
Yeah, you did. I'm going to start you again.
Okay.
Okay, off you go.
So I'm going to briefly tell you about how genome size in flowering plants can play a role in influencing their extinction risk. It turns out that the size of an angiosperm's genome can trigger substantial changes and costs that scale up from the nucleus all the way to the cell and the whole organism. And the emerging picture suggests that plants with larger genomes are less able to compete and adapt in a range of different environments. So we wanted to test if this might also be reflected in a higher extinction risk for plants with larger genomes. We were able to confirm this as we found that the larger the genome, the higher the extinction risk by as much as 40%. And so we propose, because of this, that genome size might be a useful genomic trait for enhancing plant conservation efforts because it can shed some light into the intrinsic susceptibility of extinction in plants in different environments, especially angiosperms, which is key in the face of pressing issues like global change. So for more details, please come find me in poster number 39.
Thanks. It's very interesting indeed. Thank you, Maribel. And up next we have Juliana.
Hi, I'm Juliana. I'm with the Brazilian Agricultural Research Corporation, and our work is the genetic diversity of Germoplasm banks of Vanconia speciosa, a food resource in Brazil. So this species is a priority for research in the country, so we aim to evaluate the genetic diversity of 3 Germoplasm banks. Our results showed a greater expected and observed heterozygosity, a 0.3 Shannon index, and a relatively high inbreeding coefficient that we are actually attributing to a Walland effect. So we found a low genetic diversity within all 3 banks. Then analysis of molecular variants showed a greater genetic variation within than among these populations, and a principal component analysis allowed for the separation of the genotypes according to the population of origin. So we're concluding that the knowledge of genetic diversity for the Germoplasm banks is fundamental to creating core collections for the species and sampling genotypes within biodiversity hotspots. If I may, poster 40. Thank you.
Thank you very much, Juliana. And up next we have Yuri.
Hi there, I'm Yuri from the University of Edinburgh, and this is my Master's by Research project. And I feel that I'm in a minority here because I'm with the lichens, and I'm in the Arctic. So basically my project is about lichens in the Arctic and what happens to them in the face of warming, and more specifically how warming affects lichen functioning, lichen morphology, and in more general view, how is the lichen symbiosis affected. So this is the project in the Arctic in Svalbard, warming chambers, and the lichens have been there for 10 years. So I'm looking at what actually happened to them after all this time. And come to poster 42 to see some cool microscopy pictures of lichens. Thank you.
Thank you very much indeed, Yuri. And up next we have Lorinas.
Cephalantera longifolia and rubra are endangered species in Lithuania and in most of Europe. Despite we produce a lot of flowers, we produce very little fruit. So me and my colleague made an experiment about artificial reproduction and cross and self-reproduction of those species. So 70% of the flowers of Cephalantera longifolia and 96% of Cephalantera rubra produce fruit after artificial pollination, and control was of Cephalantera longifolia 6.5%, and for Cephalantera rubra 25%. And hand pollination of flowers can be used of restoration of very small or fragmented populations of both studied species. And more of this topic and more at poster 43. Thank you.
Thanks very much. And up next, we have Diego.
Yes, thank you. Hi, I'm Diego. I work as an assistant professor at the University of Tokyo in Japan, and I'm also the curator for the TI Herbarium there. And my presentation is about that. The TI Herbarium holds a collection of 1,900,000 specimens, including 20,000 type specimens. And that's the biggest and oldest collection in Japan. That accounts for about 40% of the whole collection in the country for specimens, vascular plants talking. And there are historical and resourceful information about why the distribution of specimens in the herbaria in Japan are like that. So basically my presentation brings a bit of history and also the struggles and the strategies that we are implementing to manage that collection. So if you have any interest in knowing about that, please come to poster number 45. Thank you.
Thank you very much, Diego. And then up next we have Marguerite.
Yeah. Yeah. Okay, so I have too many slides just to say. I'm going to tell you why paleoecology and archaeology can contribute to ecosystem conservation. Well, pollinator spores can help us reconstruct past vegetation and agricultural activity, particularly livestock grazing. And archaeology, on the other hand, increases our understanding of past human settlement of the landscape. So overall, together, they increase our understanding of how humans have shaped the landscape over long periods of time. And we should care because they provide long-term ecological data that we don't have otherwise, and they also help us understand how humans have been a driving force of, ecosystem changes over a long period of time and not just the last 2 centuries. And that can also help us identify landscape management practices, fostering biodiversity, and help us understand landscape history and how to conserve, restore, or modify local ecosystems. And come see me at poster 46.
Perfect timing. Thank you very much indeed, Marguerite. And then up next we have Patrick.
Yeah, hi, I'm Patrick Weigelt, and I'm presenting the Global Inventory of Floras and Traits, or GIFT database. And in GIFT, we collect vascular plant checklists and floras. And at the moment, oh, it's gone. It's back. At the moment, we have species composition information of plants for 3,400 regions worldwide, and these regions in GIFT are linked to geographic and environmental data like island isolation or temperature, and at the species level, data are linked to functional traits which we frequently find in the floras we digitize, or to taxonomic and phylogenetic information. Yeah, this combination of data allows us to do biogeographic and macroecological analyses. And for example, this should also not have happened. For example, we recently produced global predictions of phylogenetic diversity based on machine learning techniques. We looked at past and present drivers of phylogenetic endemism and centers of neo and paleoendemism. And we looked at how different growth and life forms contribute to global diversity patterns, including also data from the World Checklist of Vascular Plants.
Oh, nearly. Thank you very much. Okay, we're getting towards the end now. Over to you, Ashley.
Hi, I'm the Data and Workflow Manager for the digitisation team here at Kew. And digitisation really got started at Kew in 2004 with the African Plants Initiative, and since then it's definitely been a project-funded effort. But in 2022 received funding to digitise all of our herbarium and fungarium collections, and we decided to go down a supplier and an in-house route. In our first year we hired 54 members of staff, as well as develop workflows across departments. We have 3 million specimen images, and 2 million of those have transcriptions. So if you want to talk things digi, I'll be up on the first floor.
Well done, thank you very much indeed. And now we have Lais. Lais, sorry.
Yes.
I nearly got there.
Hi everyone, I am a PhD student at the University of São Paulo in Brazil, and I work with the genus Zonia, which is a pantropical genus that mainly occur in the dry forest like the savannas, caatinga, and cerrado. And the main purpose of this work is to answer biogeographic questions related to the ancestral range information and evolutionary transitions between biomes, and also discuss the conservation of the dry forests where the genus mainly occur. So if you want to see my results, come to the poster number 50. Thank you.
Fantastic. And last but by no means least, we have Natalia from Royal Botanic Gardens, Edinburgh.
Yes.
Off you go.
Thank you.
Hello, I'm Natalia. I'm a PhD student at the Royal Botanic Garden in Edinburgh and the University of Edinburgh. And I'm presenting this poster on the phylogeography of Guasuma ulmifolia. Guasuma ulmifolia is a widespread species in America. So this species is widespread across different biomes, treated biomes like dry forests and wet forests and savannas. And we test the phylogeography was using target capture for morphology and geography. And my poster is number 12. Thank you.
Fantastic. Thank you very much indeed. So that is the end of the session. So congratulations to all of the speakers. A fantastic job. It really is quite a difficult challenge to get across all of the information you want to in a minute. But just to wrap up, I wanted to remind you that there's a drinks reception and poster session coming up where you can see these posters. I know which ones I'm going to. And that's in this building. You've probably already discovered where they are, but if not, ask a member of staff from Kew Gardens. This is, of course, for in-person attendees. For those of you online, I'm afraid you won't obviously be able to do that, but clearly you are able to follow up any questions with any of the speakers in your own time. Also, just to say goodbye to the online participants, and we'll be back tomorrow for day 2, and we look forward to seeing you all there. So thank you very much, and good night.